what if the next major breakthrough in cancer treatment isn't actually a single brand new chemical? What if it's a combination of ancient plants, repurposed everyday pills, and brilliant metabolic traps? Welcome to this explainer. Today, we're diving straight into the fascinating and rapidly expanding arsenal of cancer treatments. Now, if you follow oncology at all, you know we are in an era of rapid, frankly mind-blowing discovery. We're looking way beyond standard maximum tolerated dose chemotherapy. Instead, we're going to examine a totally diverse range of interventions based on some really compelling recent research, clinical data, and observational studies. We're talking everything from plant-derived pharmacology to stuff you might find in a medicine cabinet, all through the lens of how they target the fundamental core biology of a tumor.
To make sense of all this, we've broken the explainer down into 5 key areas: rethinking cancer cell death, nature's anti-cancer pharmacy, repurposing everyday medications, starving the tumor engine, and finally, evaluating the clinical evidence. Let's kick things off with Section 1, rethinking cancer cell death. So to understand these new treatments, we first have to ask the ultimate question in oncology. How do you kill a cell that just flat out refuses to die? Remember, a cancer cell is essentially just a normal cell that has completely bypassed its own self-destruct sequence. And as tumors mutate, they build up this formidable resistance to conventional drugs. So how do we get around that?
It all comes down to apoptosis. This is the genetically controlled pathway of programmed cell death. Normal cells, they naturally undergo apoptosis when they get damaged. It's like a built-in safety switch. But cancer cells actively suppress the signaling proteins that trigger this, which is exactly how they multiply indefinitely. So the primary goal of almost all cancer therapies, including the ones we're talking about today, is to force those malignant cells to flip that self-destruct switch back on.
And here is how they pull off that incredible survival trick, the Warburg effect. Normal cell metabolism uses oxygen, right? And that process naturally produces low levels of reactive oxygen species, or ROS. If a normal cell gets too damaged, a massive burst of ROS triggers apoptosis. Boom, the cell dies. But cancer cells, they actually alter their own engine. They rely on this highly inefficient, oxygen-free fermentation to make their energy. By doing this, they keep their ROS levels artificially low, totally dodging the self-destruct signal. Understanding that metabolic shift is absolutely crucial to defeating it. Moving right along to Section 2, nature's anti-cancer pharmacy.
Let's look at how naturally occurring botanical compounds target those specific pathways. Take pristimarin, for example. It's this fascinating compound found in the South American plant Espinir hesanta, and it attacks that exact metabolic vulnerability we just talked about. Research shows pristimarin actually increases those reactive oxygen species inside the cancer cells. It essentially overloads them with oxidative stress to trigger apoptosis. It's doing exactly what the cancer cell was trying so hard to avoid. And the best part, it appears to do this while protecting normal healthy cells from that very same damage.
We see something similar with compounds found in sumarote, specifically oleanolic acid. These don't just act randomly, they surgically target pathways to stop tumor cell proliferation. Studies show they lower the proteins that support cell growth and actually activate the p53 pathway, which is famous for fighting cancer and inducing apoptosis. Plus, these plant sterols help inhibit angiogenesis. In simple terms, they cut off the tumor's supply lines, stopping it from building the new blood vessels it desperately needs to feed itself. But there's a catch, right? Getting these incredibly powerful plant derivatives to the tumor safely is really difficult. Take lurdotecan, derived from the Chemtotheca plant. In early trials, it showed high systemic toxicity. To fix this, researchers use a brilliant piece of engineering called liposomal encapsulation. They basically pack the drug into a protective lipid bubble. This stabilizes the compound, lets it sneak right past the cancer cell's resistance pumps, and delivers a highly targeted payload directly into the tumor, all while sparing the surrounding healthy tissue. It's a real game changer.
All right, Section 3, repurposing everyday medications. This is where we look in a totally unexpected place, the standard medicine cabinet. Researchers are finding that common off-label drugs have potent anti-cancer properties. Let's talk about naltrexone. Traditionally, it's used at high doses for addiction, but it does something entirely different when given strictly under 4.5 milligrams a day. At this incredibly low dose, LDN acts as an immune modulator. It decreases suppressive regulatory T cells and ramps up the tumor-killing capabilities of our body's own immune system. But timing is everything. If the dose goes too high, it blocks the receptors continuously and actually reverses those cancer-fighting effects. LDN actually influences cancer progression through 3 distinct mechanisms. Beyond just tweaking the immune system,
It acts as an antagonist to specific receptors, actively throwing a wrench into cancer survival signaling pathways. By disrupting these signals, it tilts the balance back toward those pro-apoptotic proteins, literally priming the cancer cells to die. We are also seeing some truly fascinating observational data around antiparasitic drugs. There was a recent prospective observational cohort that followed 197 patients. These folks were treating a huge variety of malignancies, mostly prostate and breast cancers, with a compounded combination of ivermectin and mebendazole. And preclinical models back this up, showing these drugs can inhibit cancer cell proliferation and disrupt the cell's internal structures. So, what were the results? Astonishingly, at the 6-month follow-up, among the 122 patients who completed the survey, the clinical benefit ratio was reported at 84.4%. Just to be clear, a clinical benefit ratio means the percentage of patients who experienced either tumor regression, no current evidence of disease, or at the very least, stabilization of their disease. That is a massive number. Breaking that down a bit more, nearly half, 48.4%, reported tumor regression or no current evidence of disease. Another 36.1% reported stable disease. Now, we have to be completely objective here. These are self-reported, real-world outcomes, and a lot of these patients were combining this protocol with standard therapies like chemo or surgery. But given the incredibly high adherence rate and the really mild side effect profile, this is an area generating some serious clinical interest. That brings us to Section 4, starving the tumor engine. So traditional chemotherapy is all about the maximum tolerated dose, which, as we know, causes heavy systemic toxicity. But insulin potentiation therapy, or IPT, takes a wildly different approach. Because cancer cells are absolutely desperate for glucose to fuel that inefficient fermentation we talked about, they have up to 16 times more insulin receptors than normal cells. IPT exploits this. It uses a tiny dose of insulin as bait. The hungry cancer cells throw their doors wide open, expecting a sugar rush. And at that exact moment, a much lower, far less toxic dose of chemotherapy is rushed straight into those highly receptive cells. It is literally a Trojan horse. Other metabolic interventions try to starve the tumor by directly altering its biochemical environment. For instance, oxaloacetate acts as a glutamate scavenger, stripping away the exact fuel that central nervous system cancers rely on. We also have mineral transporters called orotates, like calcium orotate, which drives calcium right into the mitochondria. But you know what's getting a ton of focused attention lately? Lithium orotate. Orotic acid is this highly effective carrier that lets elemental lithium slip right past the cell's outer membrane with incredible bioavailability. Once it's inside, it acts as an emerging modulator. Studies are looking closely at its mechanism of action, specifically how it modulates immune pathways and actually inhibits the specific enzymes cancer cells need to survive. Plus, it offers profound neuroprotective and mood-stabilizing benefits for patients going through rigorous cancer treatments. It's a fascinating, multi-targeted compound. And finally, Section 5, evaluating the clinical evidence. With so many incredibly diverse therapies, from Amazonian plants to repurposed antiparasitics to metabolic Trojan horses, how do we separate the hope from the hype? Well, the medical community relies on a strict hierarchy of evidence. While test tube and animal studies are great for showing us the biological mechanisms, like exactly how pristinamycin triggers apoptosis, we can't just extrapolate that directly to humans. Ultimately, we have to rely on rigorous, prospective observational studies and randomized controlled trials to definitively prove real-world safety, effectiveness, and how these treatments synergize with what we already use. So, given the remarkable, almost terrifying complexity of how cancer adapts, mutates, and survives, what's our main takeaway from this expanding frontier of medicine? If cancer operates on multiple pathways at once, dodging the immune system, hijacking its own metabolism, resisting toxicity, it absolutely stands to reason that our treatments need to be just as multifaceted. The data heavily suggests that combining metabolic starvation, immune modulation, and targeted cell death might hold the real key. So I leave you with this thought. Are we finally moving away from the search for a single magic bullet and stepping into a new era of highly integrated, personalized oncology? Thanks for joining us for this explainer.